I was requested to do a talk on a workshop on statistical inference, which is kind of interesting because, speaking from my heart, I don't really like statistics, but they are useful. There was a very famous author that says that there are three types of lies: white lies, bad lies, and statistics. Having said that, there's so much we can do with statistics and biological data. So when we have numerical data to describe something, we can model whatever something that is and use a mathematical approach to describe it. That's something that we call descriptive statistics, and you are very well familiar with such approaches, like maybe measuring the average of something, determining what is the standard deviation of something, or estimating the standard error of a measurement that you're collecting. That only tells us something about what we are describing, but we can also compare the measures that we are taking to something else. Like maybe you are measuring the average height of Mexican population, and then you are comparing to the average height of people in the Netherlands. So that can tell you that there are differences in terms of genetic background, diet, environment that lead to Mexican people being relatively short and people in the Netherlands being relatively tall. And once we have such comparisons, we can do some hypothesis testing, such as trying to analyze the data that we have and see what might be the underlying factors behind the differences that we observe between two distributions. But importantly, we can also build models that we can make predictions with for future events. These are a bit, well, not complicated, but they are a bit tricky in the sense that whatever we come up with, any given model is just a prediction. We need some ways to prove it, and that's, well, particularly true for bioinformatics, as you may be familiar with the work that you do on your day-to-day basis, such as you make a prediction that these genes are associated with that function, but you still need the experimental validation to confirm your predictions. So in statistics, the observations that we make, they belong to a group, an invisible group called population. A population is something that we never really fully understand. We sample from that population, and due to technical limitations or theoretical assumptions, we are never going to fully sample 100% of any given population. Shall this population be humans, nucleotides, time, whatever? In that case, we resort to sampling from that population as much as we can, and then we model the information that we obtain from a sample, and from this modeling, we can infer some conclusion about the population, obviously with some degree of confidence or with some degree of uncertainty. When we collect the data from a sample, we can calculate the descriptive parameters, and we can model the parameters to see whether they are likely to happen or not. And when we do this, we essentially present something that is a model, and this model can be an equation, a set of intervals, probabilities, but very, very importantly, especially for biological data in bioinformatics, distributions. Distributions are something that it's very well good idea to invest time in studying distributions and how you can compare distributions. However, many statistical models assume specific distributions for any sort of data and specific numerical data types. This is something that I really wanted to fully grasp. In biology, we don't have 0.5 nucleotides, nor do we have 0.0024 proteins, or minus 1.37 organelles. also, importantly, they don't distribute normally inside the cell or inside an organism. Therefore, whenever you see someone coming up with a t-test for comparing two distributions, you probably suspect that their data might not be very well presented, because in biology, it is very rare to observe a normal distribution of something. It's more likely to observe a binomial distribution or a distribution that is skewed, and therefore you need to apply different methods to it. So that's something that we need to keep in mind. There are plenty of models. We are not going to cover all of them, because it's a never-ending, would be a course on biostatistics and not a course on bioinformatics. And for today's lecture, we are going to cover the Markov models, in particular hidden Markov models. And these are statistical models that were devised by Andrei Markov. They are statistical models that describe changing systems from one state to another. It will make sense in a bit why they are useful for sequence analysis. Before we start, how many of you are familiar or have used hidden Markov models for analysis of their sequences? Okay. How many of you are familiar with the underlying mathematical model behind the statistical, the Markov models? Okay. Okay, we have a good representation. So in any given process, we have a variable that is changing from one state to another. Once the variable is changing from one state to another, it can remain in the same state, or it can change back to the other state. And this can happen for a long time, depending obviously on the search space. In the case of sequences, it might be that one sequence is changing from one position to another, taking different states, like the residues of a DNA sequence or the residues in a protein sequence. But we are going to see later that we only need the right labels to apply the Markov models to any sort of data that comes from biological analysis. Let's cover a Markov model using a typical British traffic lights. It's not shown very well for the projector, but here is green. Just take my word for it. So in a typical British traffic lights, we know that when we are in green, it is very likely that we go to yellow, and from yellow we go to red. That's something that I found out in British traffic light. They are different from the Mexican type, because the Mexican type immediately after red goes back to green. But in the UK, they go from red back to yellow and then back to green, and so on and so forth. So that's a very simple process. We simply have a changing pattern where we can detect different states in our model. But how can we model this in a more formal way? So if we assume that our traffic lights is working properly, we know that when the traffic light is in position green, it can remain in green state, but that only happens very seldom. It is more likely, though, that it changes from green to yellow. From yellow, it can remain in yellow, but again, that also doesn't happen very often. From yellow, it can go to red, and from red, it can go, depending on the type of traffic light, it can go back to green, but that doesn't happen very often. It's more likely that it goes back to yellow, and from yellow, it can go back to green. So what we see here are the three different states for our traffic lights, and the numbers that we see, and the arrows that we see connecting the different states are the transitions explaining the model. The numbers associated with each arrow correspond to the probabilities of each transition. That is something relatively straightforward. There is not so much to think because, well, we see that the probabilities of transition here are high. And essentially, this would be the formal representation of the same thing. So what if instead of taking a traffic light, we take a nucleotide sequence or a protein sequence? Instead of having three different states, we can have four different states, or maybe we can have 20 different states for amino acids, or maybe we can have different states that they don't necessarily have to deal with the nature of the sequence. It might be associated with the function of the sequence, whether is this part of the protein binding a metal ion? Is this part of a genome sequence coding for a protein? Is this part of a genomic sequence being present in multiple copies, and so on and so forth? We basically just need the right labels for every category. Now, a Markov model needs to fulfill certain properties. It can be of any finite length. When it comes to the traffic lights, well, we don't really see a start and end because we assume that this is going on forever until the power goes out. But in reality, especially for Markov models applied to biological data, we assume that they have a finite length. They do have start and end states that are not present in the model that I just shown. And a transition occurs when the system moves from one state to another. The states we are going to cover later on. But probably one of the most important parts of a Markov model is that it's memoryless, meaning that the transition from state B to state C depends only on the current states, in this case, state B. That is, whatever happened in state A doesn't really matter for the transition for B to C. All the states in the model are observed, and if we cannot observe them, we assume that their probability is 0. But that's a Markov model, and Markov models are not particularly widely used in bioinformatics. So just for a bit of fun, you can explore this website, and we can explain how different Markov models look under different assumptions, and how can we play with these models. So if we go to the middle part, we will see here just a ball changing from one state to another. And in the next list of lists, we will see the probabilities of transition from one state to another. And basically, we have 3 lists, one corresponding to each of the states, and the transition to neighboring states. So for instance, in this case, we have A, B, and C, probability of transition from A to B, and A to C. State B, transition B to A, B to B, and B to C. And state C, transition of C to A, C to B, and C to C. And then you can play with the numbers to see, well, also, importantly, the arrows are as wide as the probabilities. So if you set, for instance, this one to 0.1, and also this one to 0.1, and this one to 0.8, you're going to notice an increase in the size of the arrow. So you can play here with the transition, the probability of transition between one state to another, and see how the system behaves. Obviously, this is a bit silly because we are still not describing anything that is biologically relevant, but it helps to understand how a Markov model works in terms of the transition probabilities. The developers behind this project, they provided 3 interesting examples. So example 3 is the one that we are just seeing, but if we load, for instance, example 2, we have a collection of multiple states, which is resembling more what we can observe in biological sequences. So in biological sequences, you don't have only 3 states. You have multiple states corresponding maybe to the different residues, or maybe corresponding to the length of any given sequence. And as you can see from the matrix presented in the box, the model now becomes more complicated. In this case, we are assuming uniform distribution of probabilities. So this particular Markov model describes how the state can go from one position to another, almost by random, because all of the transitions have the same probability. So that's a very cool website, because then you can play with it and establish your own model. So it would be, well, a good idea for you to try. Importantly, in this case, all of the changes are permitted depending on their transition probability. However, because of that, whenever we increase the number of states, we are going to increase exponentially the number of combinations of different paths. That is, if we have 3 states, we can only have 6 combinations. If we have 4 paths, then we can have 10 combinations, and so on and so forth. And now back to our explanation. Okay. But what happens that when we don't really see what is going on? In this example, we have this lady here, and this lady here is looking in this direction, but she's not looking at the traffic lights. Do you think she can tell whether the traffic light is changing from one color to the other? Yes? No? Why not? Why yes?
She can look at the cars.
She can look at the cars. So even when she's not directly observing the traffic light, she still can see the car is passing, because whenever this car is passing at this point, she will notice. And when she notices that the car is passing, she will say, okay, so the traffic light is potentially green. If the car stops, then she can say, potentially the traffic light is red. That's not necessarily the case. In this scenario, we have the same traffic lights, and the probabilities of the model are the same in terms of the transition probabilities. However, we now have another set of probabilities. We have the emission probabilities, that is, things that we can actually see. In this scenario, we are not seeing at all the traffic lights, but we are seeing whether the car stops or whether the car keeps going. Okay? So we have basically 2 categories, whether the car stops or whether the car advances. And here we have the probabilities of emission for any given state. So if we are in state red, we are expecting the car to be stopping at least 85% of the times. But, well, you know, not everyone respects the traffic lights. So there might be a chance that the car is passing, but the traffic light is still red. In green and yellow is different. Basically, when we see green, there is no reason for us to stop. So 99% of the times, whenever we see green, we keep going. And the same is true for yellow. Actually, in yellow, I think people tend to accelerate a bit more. So once we have this, then we have a hidden Markov model. That is, we are describing something that is changing from one position to another without actually observing directly the states. And we can describe this by as a group of matrices that will tell us essentially how this is behaving. So for a hidden Markov model, we have some ingredients. We need a list of states that are hidden. We are not observing such states. We need a list of initial probabilities that in the toy example we didn't show because we assume that this is going on forever, but in reality we need a start and stop of the hidden Markov model. We need a matrix of transition probabilities, which we already have from the initial model, but we also need a matrix of emission probabilities. This will become clearer when we go to the practical. And importantly, because this is still a Markov model, this needs to be memoryless. That is, whatever happened in the previous state doesn't need to affect or won't affect the transition to the next state. So that's for traffic lights, but what about bioinformatics? In many cases, we just need to label our data the right way. Whenever we read a sequence by a massive sequencing or any technology, we only see the sequence, and that's something that, well, as the name implies, we can observe, but we don't know what's inside those sequences. So if I present you a sequence, any given sequence, can I ask you, well, is this region coding for something? Is this a regulatory property of the genome? Is this something that is methylated? Is it involved in, I don't know, functional motif of a protein? So those are the hidden states in bioinformatics. The things that we are seeing are the sequences. So let's play with a toy example of a splice site. How many of you work with eukaryotic genomes? Okay. So in many eukaryotes, the splice site is given by the consensus sequence, GT. All of the, or at least the canonical splicing sites, they all start with G. And they can be followed either by T in the majority of cases, or with A in a minority of cases. So we can describe a hidden Markov model as follows, such as we have the sequence, and in principle, we don't know whether this is an exonic region or it corresponds to 5' of a splicing donor, and the next part corresponding to an intron. What we can do is build a hidden Markov model where the transition probability from exon site to exon site is close to 90% because we are expecting most of the sequence to be coding. And we can assume for exonic regions that the frequencies of the nucleotides are homogeneous. However, whenever we reach a G, there might be a transition probability associated with the start of a 5' splicing site. In this case, we have the majority of donors starting with G, and on occasion, A. Depending on what we see next, we can conclude whether this is a proper splicing site or if this is just another exon. And depending on the transitions, we can come up with a conclusion that this is actually the splicing site for removing an intron in the processing of a messenger RNA. Now, when we have an observed sequence in a pre-existing hidden Markov model, we can calculate 3 main problems, or we can use our hidden Markov model for 3 main problems. One, we can calculate the probability that the hidden Markov model that we have here is actually producing the sequence that we are observing. That is, we can go back and forth, challenging the sequence against our hidden Markov model and determining the probability that this is coming from random chance or from the hidden Markov model actually predicting the sequence. This is useful for prediction of functional domains, and we will see in a minute how it works. We can also calculate the optimal state sequence that explain our sequence. And for this, we are aligning our sequence to the hidden Markov model, as it is in our example. But we can also infer and optimize the parameters of the hidden Markov model. So even when we start with this set of assumptions, we can have a ground truth, a collection of ground truth sequences, and then retrain our hidden Markov model to come up with better estimates for the transition probabilities and the emission probabilities. So let's have a very quick example. In this case, we have a hidden Markov model where we have a start position. We don't have an end position, but that'll come up later. And we have 2 types of categories. We have a category for high GC content and for low GC content. So in high GC content, we see that the emission probabilities for C and G are relatively high, whereas in low GC, the emission probabilities for A and T are high. In this case, from the start, we have that the transition probability for start to any of these 2 is equal, because we are not assuming anything. And once we are in the high state, we can either remain in high state or change to low. We don't really have a transition to here because, well, in bioinformatics, the hidden Markov models tend to be quite directional. In other cases, you can have backward transitions, but not in this case. And so basically, we have the same transition probabilities for being in low GC and remain in that, or change to high, or being in high and remain in high, or being in high and change to low. Now, that's our basic model, but let's challenge a sequence. In this case, we have a toy sequence called GGCA. Just by looking at the sequence, we can already see that it's high in GC, meaning that it's very likely that the model is actually predicting the sequence. But let's have a look at why this is true. So in Markov models, in hidden Markov models, what we need to do is essentially reconstructing the path of probabilities, giving rise to any of these residues, the ones that we are actually seeing, and trying to infer what is the probability of them belonging to all of these hidden states. In this case, we define the probability of G being part of H as 0, in this case, because in the start position, we don't really have any probability of emission for neither A, C, G, or T. And then we add the probability of the emission probability, the transition probability from start to high, 0.5, times the emission probability of G under the assumption of high GC. So when we add this product, we have 0.2. And that is for the probability of G being emitted for under H. If we calculate the probability of G being emitted by L, we have 0, the probability of G emitted by start, which is 0. And then we sum the probability of transition from start to low. And then we multiply by the emission probability of G given low, in this case, 0.1. And then we move on across the sequence. And then what we need to do is take the previous probability and keep adding on the probability of this nucleotide being emitted by this particular state. So we already have the 0.2. But we need to multiply by 0.6. Oh, there must be a typo here. Need to check that. And then we multiply by 0.4, which is the emission probability of, oh no, never mind. So this 0.6 is the transition probability of remaining in the category of high. And then we multiply by the emission probability of G under high. So this previous probability times the probability of remaining in the same state, or a transition of high to high, and then the probability of emission of G under this one. And we have this number. And the same is true for G being emitted by the other category, and we can progress in the alignment calculating the probabilities of each nucleotide being emitted by these states, and when we reach the end, we add the two probabilities that we came up with, adding up to 0.0051. And then you may think, well, this is boring. Why is this relevant? Because if we assume that under normal conditions, the 4 nucleotides would be equally distributed, we will see that the probability of A in the background distribution, it will be 0.5, probability of C, 0.25, G, 25, and T, 25. Because we have a sequence that is 4 nucleotides long, we can multiply the corresponding probabilities of all the nucleotides, and then we will see that this is a probability of 0.0396. What is the relevance of this? Our model has a higher probability here, meaning that this sequence is not happening by chance, meaning that the hidden Markov model is actually predicting that the sequence is not happening by chance. Now, this is a very simple example, but imagine that you have a more complex model where you have as many states as you have length in the sequence corresponding to a functional site in a protein. You can build your model, you can calculate your probabilities, and from there you can determine whether the presence of a series of nucleotides in a protein corresponds from something happening by chance, or if it's explained by a functional part of the protein. Now, questions so far? You sure? It's now or never. Okay. Remember, this class is also memoryless.
May I ask a little question?
Sure.
Like if we return back to the traffic lights example,
Yes.
what is actually like a step? Like, because there is like, because it's a continuous process. How
Yeah.
would we define in a, well, if we have a process that is continuous,
Yeah.
and not discontinuous like with the nucleotides, how would we actually, this probability, is the probability per some time or?
Well, in bioinformatics, it's very helpful that the data is already discrete. So importantly, I think the only type or the most popular type of Markov models applied to continuous model are Gaussian mixture models of hidden Markov models. But I'm not sure if they are very used.
I think I can answer
Yeah.
this question. So you're asking about like, because this is in continuous time.
Yes.
Yeah, and it's like a okay, so the thing we're looking at right here is actually like a simplification where we believe that time is on a grid. But it's like, you just count, okay, so in this second, we have exactly this state, and in 1 second, what will the state be? There are like variants where you let time run freely, but that's not what we're talking about here. So it's to connect it with the discrete nature of sequences.
Okay, yeah.
And also, importantly, for applying this to bioinformatics, we are also under the assumption that two states might be mutually exclusive. That is, a sequence here. No, sorry. 1 second. A sequence here, which is not all the time true, doesn't have to be all the time, or the splice site needs to be the splice site. It doesn't have to be a part of an exon. There are some violations to this, like in alternative splicing, where actually the donor is part of an exon. But in regular conditions, we assume that two states might be mutually exclusive. Okay.
I'm sorry.
Yep.
Can you move to the next slide? Next. I'm not really sure I understood how 00396.
You multiply this for this, for this, for this. Let's see if it holds. I mean, it doesn't hurt to double check. 0.25 times 0.25 times 0 times 0.25. Yep.
We don't know the optimal number of hidden states, right? It's not always, right? Like for plotting models, you don't know the number of states, right?
That's a very good question.
The hidden states will not be.
That's a very good question. Suppose that you have multiple sequence alignments, and for this multiple sequence alignment, you take proteins from human, plants, insects, and the functional part of the protein basically falls in this region where these residues highlighted in boxes correspond to the functional part of the domain. Because we have a variable length in the sequences, is exactly as you say. We don't really have the exact number of hidden states, but this is a self-limiting problem. Why? Because the more we have, the less we have representation here or here, the probability will decrease here and here. So there are some algorithms that essentially say whenever we are modeling the hidden Markov model, we can trim whenever we reach, whenever the probability here is not enough to be considered part of the.
So in this metaphor that you're using, women can actually figure out that there are 3 lights and not 2, for instance, right? But you could also use
that is true, yeah, because in the example, you're absolutely right. we only have two emissions, which is either the car goes or stops, but the probabilities here are almost the same. So for the lady, these two states are almost indistinguishable.
So my question is, there is a way to kind of pick the optimal number of states for your model, right?
That's where we're going, actually. The next problem that we can do, that we can solve for this is, given any sequence, in this case, this toy example here, but this can also be related to the car problem, how many cars we see passing after us, and we are trying to infer the state changes in the traffic lights. This is a factorial problem. So we have two hidden states here being high and low, and we have 10 different positions here. So any path can explain this sequence from all of the transitions being it started in low and remain in low and remain in low and remain in low, to it started in high and remain in high, 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 high. Or maybe even something alternating like it started in high, then low, then high, then low, then high, then low. So there are approaches for you to model this, and they are actually very similar to the forward alignment here, where we are calculating the probability of a sequence being generated by the hidden Markov model. What we do is that we calculate the probability of a given path. In this case, let's suppose that we only have three nucleotides, and we might model the path. It started from the L states, then it changed to high, and then it changed to low. This is a very likely scenario, but we don't know if this is true or not. So what we can do is calculate the probabilities giving rise to that particular path. So the first transition would be from the start position to low times the probability of emission of the first nucleotide in the sequence given the low category times the probability transition from low to high, this one here, low to high, times the probability of residue G, second position, under the high category, times the transition probability from high to low, that is this other one here, times the probability of the third nucleotide under the low label. Because we already have those numbers, we simply can calculate all of these products, and then we can calculate the probability of this given path. That is easier said than done, because at least for this example, we have 1,024 paths. And whenever we increase the number of nucleotides by 1, it becomes exponentially growing. So if we make use of the Markov property of the model, that this is memoryless, like it doesn't depend on the current state only affects the next one, we can do a backtracking similar to dynamic programming that we do in sequence alignment to calculate what is the most likely path to model the sequence. That is called the Viterbi algorithm, that we won't explain too much in detail because we are actually going to build the Viterbi algorithm in the practical, so we can hold your horses. So why hidden Markov models are popular in bioinformatics? They have been described as the Legos of bioinformatics, and they are extensively used in ab initio gene predictions such as Glimmer, GeneMark, and Prodigal, and classification on proteins and evolutionary biology. And also because they are very efficient in terms of hardware requirements, they have very low memory footprint. And remember what I told you about using the right labels. So far we have been describing sequence states, but it doesn't have to be sequence. So whatever we can put a label on, we can model as a Markov model, like these examples, for instance. In this case, we have sequences. Yes, they're still sequences, but we do have labels of a sequence being under positive or negative selection, and we can model whether they are important or not for any given function, especially in the context of population genomics. We can also do comparative genomics using hidden Markov models. And we can also model the transitions of whatever we want. In this case, we have microbial genomics. We can encode everything as presence, absence. We have immune systems in prokaryotes using Markov models. But this one caught my attention because it's actually quite easy to, well, not quite easy, but relatively straightforward to implement, which is detecting the copy number variation in microarrays, because you can model the transition of one particular region of the genome being present in 2 copies, that is 1 copy per parental line, but every now and then you have either a complete chromosome duplicated or being in multiple copies, such as in Down syndrome, or particular regions of the chromosome being duplicated, like maybe coming from uniparental disomy or so on. So you can model the number of copies at any given state in the chromosome, and then model what are the transition probabilities for each state, and say, okay, this stands to reason that this belongs to a region that has high copy number variation, and therefore we can make a conclusion on the potential effects of this copy number variation of this particular region. So all this being said, don't look at the sequences just as sequences. You can put label on the sequence, and then you can do Markov modeling. For instance, you can. There is this group of organisms that have almost all of their sequences in a single coding strand. So in many organisms, you have gene in forward, then gene in reverse, then gene in forward, then gene in reverse. But the trypanosomatids, they do have their genomes, almost all of the sequences in forward. So you can model these transitions to obtain a meaningful conclusion, such as what are the regulatory mechanisms behind the transition between a cluster of genes that go in one direction and a cluster of genes that go in another direction. And this doesn't have to do anything with sequence at all. So all the data that you have, it's very likely to be categorical or at least discrete. And if that is discrete, then you can do Markov modeling with it. And I hope you enjoyed. I think we are good on time. So, yep, thank you. Questions?
I might have one question.
Sure.
So, my understanding and what I know about the Markov models is that we need to have a defined structure when we start. Is there a way to search for an from the data from generate the chains? Right?
Yes. In the practical, we have 2 examples. One that I can think of. Initially, you can start under the assumption of equal distribution of frequencies. And then once you start challenging your Markov model against the real data, then you can readjust the parameters to change both the transition probabilities and the emission probabilities. But you can also start with pre-aligned data, such as you have a collection of proteins that you know they have the same function. So even when there is some degree of divergence in those sequences, you can still infer that the transition probability for state A to state B, state A being this residue is important for the function of the protein, and state B being this is not relevant for the function, you already have that information from the multiple sequence alignment. So this is one way for you to give the model a structure from the beginning. In another example, you can train the coding sequences from an organism, and we will see this in the Glimmer practical, where even when you don't have the sequences aligned, you still know what are the transition probabilities, because you know that they are the ground truth sequences that code for something. And then you can recalibrate your model based on frequencies of nucleotides. For instance, under equal frequency distribution, you can assume that anything can be a start codon. But once you get the actual open reading frames, you will see that, well, in the majority of cases, you have ATG at first. So you already have one transition probability from start to ATG being close to 90-something%. Yep.
Hi, I just wonder, are there any methods to bypass the message to the model that chromosome is not like a total, so it does have like a very, very, very big length, which is, and the chromosome has a start and an end for the macro model? Yeah, because like we have a whole genome, yeah, but we also have separate chromosomes. That's actually the question I'm trying to solve now, like how to bypass this message. This is the start, this is the end, and this is another chromosome.
I see.
Do you need them to train one model per chromosome, which is not also feasible?
I see what you mean. Yes.
That's a good
It's a, well, I did something in the past for that. So in my case, I was interested in the organization of genes per strand. So I did separate the chromosomes, but in the final model, whenever I reach the end of a chromosome, I reach the counter, reset the counter for starting again in the next. Well, in my case, I was not using probabilities, but that can be implemented as well. Because then you can have like a variable length model. So you have one start and end for big chromosome, one start and different end for another chromosome, and so on and so forth. And importantly, when we see the structure of hidden Markov models in PFAM, there's also probability of premature termination of the model. So yes, you can do it.
Just technical, so for a model that you don't know the probability, you would start with total randomness, right? So 0.5 for 2 cases, like 0.25.
Yeah.
And how much would you have to train to converge to an actual model?
I think
because
that can.
It changes, like.
Yeah. I don't think. Well, I think it requires. It depends in two things. Let's say gene finding, and let's have two examples. On the one hand, we have, I don't know, let me think. Intracellular bacteria, like maybe Salmonella. Low GC content, relatively small genome-ish. And on the other hand, you have Mycobacterium tuberculosis, still intracellular, but high GC and relatively big genome. So if you start using the same probabilities for both, your model will not be performing well because this one doesn't have, equal distribution of frequencies, neither this one. But after the first round of training, the probabilities change quite drastically if you provide the right number of training genes. So in the case of Glimmer, when you extract the ORFs that you know are true, you can train like maybe with 100 genes, and those would be enough to recalculate all the probabilities, and they will change drastically, actually. So I don't think it needs that many iterations for recalibrating the. But you also run the risk of overfitting.
Yes, that's what I'm thinking. And would you have to account, for example, for the higher GC content? Like, are there other things you would take into account?
Usually you do. So there are pre-computed models that you can start your search with, and it is advisable for you to start with something rather than recalibrate and then let the algorithm do its thing. Yep.
Any other question?
All right, then we have a break, right?
Yes.
Woo, coffee.